Hello, my name is Joe Keating. I'm a postdoctoral research associate at the University of Bristol and part of the Paleobiology Research Group. I'm going to be talking to you about morphological evolution. Over the next 3 short lectures, I'm going to try and answer some specific questions related to solving the problem of morphology. By the end of these lectures, hopefully you will have a good understanding of the benefits and limitations of different methods for estimating phylogeny from morphological data, as well as some of the challenges in analysing morphology and understanding its evolution. In this lecture, I'll introduce the main ways of estimating phylogeny using morphological data and explain how they differ. As paleontologists, we are interested primarily in the fossil record. Fossils provide evidence of life on Earth over millions of years. Yet, in order to truly appreciate the significance of fossils, we must place them in a phylogenetic framework. Phylogeny gives context to our fossils. It allows us to establish taxonomic classification systems, to understand how traits change through time, to characterize the evolution of function, to rationalize stratigraphic range data, to reconstruct the evolution of ecology, and even allows us to explain taphonomic differences between groups. The fact of the matter is that without the context of phylogeny, paleontological data is meaningless. Historically, the placement of living and fossil taxa in phylogenetic trees was informal and based on the intuition of individual scientists, using the stratigraphic ages of the respective taxa and a few morphological characters that were considered important. In the 1960s and 70s, a new method became increasingly popular. Cladistics is a method of phylogenetic estimation based on shared derived characters. It paved the way for formalized phylogenetic estimation methods. In order to conduct phylogenetic estimation, we require a source of character data. Today, most phylogenetic estimation is conducted using molecular characters such as DNA or amino acids. However, the vast majority of organisms that have ever existed are now extinct, and all we have left of them is a few fossils. So estimating phylogeny using morphology is still as important as ever. Typically, we use discrete morphological characters, such as the absence or presence of a particular trait, or else the condition of a trait. There are a number of competing methods for estimating phylogeny from discrete morphological data. Cladistics uses the principles of maximum parsimony. This is also known as Occam's razor, named for William of Ockham, which states that the simplest solution is the best. More recently, probabilistic methods have been applied to discrete morphological data. These include maximum likelihood and Bayesian inference. Bayesian inference is named for the Reverend Thomas Bayes, pictured on the right. Ronald Fisher is widely accredited as the man responsible for popularizing maximum likelihood. I've chosen not to include his portrait here due to his problematic views on eugenics and race. Let's have a closer look at these methods, starting with parsimony. Here we have some discrete character data for a group of tetrapods. We can map the characters to a particular tree topology and count the number of state changes. This example tree requires 6 character state changes. Following the principles of parsimony, our preferred solution is the tree that requires the least number of character state changes. Here are all the possible unrooted tree topologies for our taxa. Go ahead and pause the video here and see if you can identify the most parsimonious solution. Here are the number of character state changes required by each tree topology. We find that this tree requires only 3 changes and is therefore the most parsimonious tree. And indeed, when we map the characters to the tree, we see that only 3 state changes are required. In standard parsimony analyses, all characters are given equal weighting. Implied weighting is a variation of standard parsimony, in which characters with less inferred homoplasy are given more influence in the analysis. In contrast to maximum parsimony, probabilistic methods require a model of character evolution. The most widely used model is the MK model. The MK model describes the evolution between K states. Here we have a 2-state model, figure A, a 3-state model, figure B, and a 4-state model, figure C. Although they share the use of a model of evolution, there are important mathematical and philosophical differences between maximum likelihood and Bayesian methods. Likelihood is defined as the probability of observing some data given a hypothesis. To use a phylogenetic example, likelihood equals the probability of observing some discrete character data given a tree topology and model of evolution. Bayes' theorem is a bit more complicated. It states that the posterior probability that a hypothesis is true given some data is equal to the likelihood times the prior probability of a hypothesis divided by the probability of observing the data under all possible hypotheses. Let's break this down a bit. In order to have a high posterior probability, a hypothesis must have a high prior probability, i.e. fit some pre-existing data that we know about the problem, and have a high likelihood, i.e. fit the new data that we are trying to analyse. The use of prior information is the biggest advantage and biggest disadvantage of Bayesian inference, and is the key difference between Bayesian and maximum likelihood methods. Priors can be very influential in Bayesian analyses. Consequently, inaccurate priors can lead to inaccurate posterior estimates. However, often we can make reasonable assumptions that help us address particular problems. Bayesian allows us to incorporate these prior assumptions directly. To use a classic example, without any prior knowledge, you might calculate the probability of successfully navigating an asteroid field as 3,720 to 1. But with the prior knowledge that your pilot is Han Solo, the best pilot in the galaxy, your odds will drastically improve. A problem common to both parsimony and probabilistic estimation methods is tree sampling. As the number of tips increases, the number of possible unrooted trees increases vastly. The vertical axis in this plot is a log scale, and to put into context quite how big these numbers are, above this line is greater than the number of atoms in the known universe, and above this line is greater than the number of atomic vibrations in the history of the universe. So, as you can see, the number of unrooted trees quickly surpasses the number of things we can count in a material universe. Clearly, we cannot measure the parsimony or likelihood score of every unrooted tree, so instead we need to sample tree space using algorithms. Parsimony analyses typically utilise heuristic hill climbing algorithms, which tend to sample one or a few most parsimonious trees. These trees can be summarised using a majority rule consensus, a tree that contains bipartitions found in at least half of the most parsimonious trees, or a strict consensus, which only contains bipartitions found in all most parsimonious trees. Maximum likelihood methods also use heuristic algorithms to sample a single tree with the maximum likelihood score. In contrast, Bayesian inference utilises the Markov chain Monte Carlo, or MCMC algorithm. MCMC typically takes thousands of samples and, importantly, trees are sampled in proportion to their posterior probability. Consequently, areas of tree space with high posterior probability are sampled more frequently. One of the benefits of Bayesian MCMC sampling is that it provides an intuitive way of assessing topological support. We typically summarize Bayesian posterior trees using a majority rule consensus. Because trees are sampled in proportion to their posterior probability during MCMC, the frequency at which a bipartition occurs in the posterior trees is proportional to its posterior probability. Assessing support for parsimony or maximum likelihood trees is less straightforward. We can use data resampling methods, such as bootstrap resampling, to gauge support. Under bootstrap resampling, we create a replica of the original data matrix by randomly resampling the characters. We then use this resampled data to infer a tree. If we do this thousands of times, we can estimate the support of bipartitions in our original tree by the frequency they occur in our bootstrap replicate trees. We can then collapse our original tree so that it only contains bipartitions with over 50% support. Different inference methods and search strategies produce trees with very different levels of precision. This will become very important in the next lecture when we try to determine which method is most accurate. To summarize, here are the parsimony, maximum likelihood, and Bayesian methods compared. Both maximum likelihood and Bayesian inference are probabilistic methods that make use of a model of character evolution, usually the MK model. In contrast, parsimony methods have no underlying evolutionary model. They rely on the principle of Occam's razor. Parsimony and maximum likelihood typically use heuristic hill climbing algorithms that sample one or a few trees. Bayesian, on the other hand, uses Markov chain Monte Carlo to sample thousands of trees in proportion to their posterior probability. Parsimony methods output the most parsimonious trees. Maximum likelihood finds the single tree with the maximum likelihood score, and the Bayesian MCMC algorithm produces a posterior sample of trees. Most parsimonious trees are typically summarized using a strict or majority rule consensus tree or a resampling consensus tree. Maximum likelihood analyses typically report the maximum likelihood tree or a resampling consensus, and Bayesian methods typically report the majority rule consensus tree. You can find further information about parsimony and probabilistic methods of phylogenetic estimation in these sources.